Joining us today. My name is Dr. Chris Moore. I am the Senior Advisor for Public Health Science and Policy at CIDRAP, and I am the moderator for this webinar. The focus of today's briefing will be on discussing the recently launched coronavirus vaccines research and development roadmap, or what we call the CVR. The CVR link will be posted in the chat shortly for your information, and I do encourage you to take a look at the whole document. Before I introduce the speakers, I'd like to give just a brief overview of the CVR and the process that we use to develop it. So first, the purpose of the CVR is to provide a comprehensive, cohesive framework and timeline to accelerate R&D of broadly protective coronavirus vaccines that are suitable and accessible for global use. The CVR was funded by the Bill and Melinda Gates Foundation and the Rockefeller Foundation, and we want to thank them for their generous support of this effort. The CVR was generated through a collaborative and iterative process that was led by CIDRAP at the University of Minnesota. For this process development, we first did a fairly thorough literature review to identify gaps and barriers and to create an initial draft of the roadmap. We then engaged 50 scientific experts from around the world to serve as part of a CVR development task force. These experts provided commentary, review, edits, and revisions to the roadmap. We also had a public comment period last fall to solicit additional input from a broader group of stakeholders. The roadmap was then published in February of 2023, and we also published an article about the roadmap in the journal Vaccine at that time. The roadmap is organized into 5 major topic areas. The first is virology applicable to vaccine development. The second is immunology and immune correlates of protection. The third is vaccinology. The fourth is animal and human infection models for coronavirus vaccine research. And the last one is policy and financing. Each topic area contains an overview of key barriers and knowledge gaps, and then time-bound goals and milestones to address each of the issues. We also included some additional research priorities for consideration. So that gives you just a little bit of a sense of the roadmap and how we created it. And now I would like to briefly introduce each of our speakers who have so graciously agreed to join us today. I'll introduce the speakers in the order that they will be speaking, and then I'll turn it over to the first speaker. So our first speaker is Dr. Mike Osterholm. He's the director at CIDRAP at the University of Minnesota. He's an epidemiologist by training, and he has had decades of experience advising the US government and international bodies on pandemic preparedness and how to address infectious disease threats. He's the author of hundreds of papers and the author of a New York Times bestseller, Deadliest Enemy. Dr. Linfa Wang is our next speaker, and he's a professor and is the director of the Emerging Infectious Disease Program, Duke NUS Medical School in Singapore. He is also the executive director of the Program for Research in Academic Preparedness and Response in Singapore. He's one of the world's leading experts in zoonotic diseases, bat immunology, and pathogen discovery. Our third speaker is Dr. Dan Barouch. Dr. Barouch is the William Bosworth Castle Professor of Medicine and Professor of Immunology at Harvard Medical School, Director of the Center of Virology and Vaccine Research at Beth Israel Deaconess Medical Center, and part of the Bill and Melinda Gates Foundation Collaboration for AIDS Vaccine Discovery. He's a physician, an immunologist, and a virologist known for his work on the pathogenesis and immunology of viral infections and the development of vaccine strategies for global infectious diseases. Our next speaker is Dr. Charlie Weller. She's the head of prevention infectious diseases at the Wellcome Trust in London. Since 2016, she has been leading a team to develop new and improved vaccines and antibodies, gaining a better understanding of protective immunity and strengthening the connection between research and decision makers. Our final speaker is Dr. Nahid Bhadelia. She is the Senior Policy Advisor for Global COVID-19 Response and the policy lead for Project NextGen on the White House COVID-19 response team. She is a board-certified infectious disease physician and associate professor of Medicine at Boston University School of Medicine, and the founding director of Boston University's Center for Emerging Infectious Diseases Policy and Research. So we'll have time at the end, after all of the speakers have stated their remarks, we'll have time for questions at that point. So if you have a question during the briefing at any time, you can enter it into the Q&A function on Zoom. Please enter your name, your organization, and your question. We hope to cover as many questions as possible. With that, I will turn it over to our first presenter, Dr. Michael Osterholm. Mike?
Well, thank you very much, and welcome to friends, colleagues, people who will be friends in the future, and colleagues in the future who will help us respond to these infectious disease challenges. Let me start out by first saying I want to thank Dr. Moore for her incredible efforts overseeing this project and bringing together all these international experts and the steering group, as well as the 11 CIDRAP staff that poured their hearts and souls into this project for the better part of a year. Thank you. Your leadership has been invaluable. Today, I just want to set out what I think is really the framework for how we look at this issue. I've been writing about and concerned about pandemics dating back into the 1990s. And in fact, now have realized two separate pandemics, 2009 with H1N1 influenza, and of course now SARS-CoV-2 and this coronavirus pandemic. This is not the last of the pandemics that will confront us. And given a whole lot of demographic and human contact, animal contact issues, agriculture, world population, we can expect to see the frequency of pandemics potentially increase. And some of them could be much worse than we've seen to date. Must we remember that the 1918 influenza pandemic, should it have occurred today with similar numbers, would have been many, many millions more individuals dying than actually have died with SARS-CoV-2, which in of itself was a disaster. On top of that, we know that there could be future coronavirus that emerge that could cause even more challenging pandemics, ones that have more of the virulence of a MERS or SARS-like virus, but with the transmissibility of a SARS-CoV-2. So it's critical that we prepare our world for these possibilities with the most important tool that we can know: effective, safe, and durable vaccines. And so it was actually a year and a half ago that I kept thinking about what we were experiencing with the SARS-CoV-2 pandemic and the fact that we had concerns about a very good vaccine saving millions of lives, but not what we actually would have hoped we would have in terms of durability, ability to respond to different variants, avoiding immune evasion, etc. All the things that we know so well. And at that point, we said, you know, we can do better. We were at that time leading the world with the influenza vaccine roadmap that Dr. Moore also played a very key role in helping to develop. And we understood that this would be a long, laborious process. If someone said to you today, from a biologic standpoint, we want you to create the biologic World Trade Center towers all over again, but we want them done in 2 weeks, no amount of money would have been enough to make that happen. It takes time. It takes research. It takes development. And so when we laid out this roadmap, we laid out with the input of these 50 experts that Chris talked about, who were absolutely critical, of which Dan and Linfa were very important parts of that. Charlie also, as part of the steering group, were part of that. And we tried to be aspirational at the same time realistic. And so we knew that it was not going to happen overnight, that we would see potentially a game-changing coronavirus or even the influenza vaccines, but we had to start. And this is a remarkable start to say that we now know what are the questions we have to answer before we get to the second set of questions or the third set of questions. What is it that we need to achieve if we're to create a vaccine for a world that might even be deployed before the next pandemic and prevent it from ever emerging? Or when the next pandemic emerges, that vaccine is readily available, highly effective, and wouldn't it be great if it had the protection of a vaccine like the measles vaccine, 2 and out? And so today we're here to talk about this possibility. And what I'm very excited about is we're here to talk about what's possible. And we're going to hear about the science. We're going to hear about the concerns of what might happen. And we're here to hear from the White House about their efforts to make sure that we don't forget that the pandemic might be on its waning days, we hope, but that it's not the end of pandemic threat. And I would just close by saying, for me, at this point in my career, after almost 50 years in the business, what is it that gets me up in the morning? What is it that makes me want to continue to be challenged by these infectious agents? And I can't think of a more meaningful, a more important, and frankly, more gut check opportunity than to help create game-changing coronavirus vaccines for my kids and grandkids. That's enough to keep me going every day. And it should be enough to keep all of us going for our kids and our grandkids. So today you're going to hear the science behind that, you're going to hear the policy behind that, and you'll understand better what in fact this roadmap represents. So thank you for being with us. Know that this is the beginning. This is not the middle, this is not the end. This is the beginning, but it's a great beginning to work from. So again, thank you to our experts, to Dan, Linfa, Charlie for your participation. Ahid, thank you very much for being here from the White House. And with that, I'll turn it back to you, Chris. Thank you very much.
Thanks, Mike. And thanks for giving us that perspective. Great perspective. And thank you for offering that. Now we'd like to turn to Dr. Wang.
Thank you, Chris, and thank you, Mike. And so first of all, really congratulations for your team for a fantastic job. So my job is really now to introduce, to develop a better vaccine, we have to know the enemy, right? The enemy is our viruses. So I just want to give you a brief introduction of the coronaviruses and their natural reservoir and our current surveillance, effort and what we can do better in future. So start with the virus, even taxi drivers, I go to they're expert on coronaviruses, but I think, yes, everybody knows about coronaviruses, but this is the largest single-stranded RNA viruses. So actually, because the large RNA genome nature make them also very mutatable, and they're very prone to a recombination. Currently, the family has 4 different groups or genera. We name them alpha, beta, and gamma, and delta. Beta is the most sort of a risk for human because they are capable of infect human and cause severe disease. So the 3 zoonotic coronaviruses are all in this beta genus. So we have the SARS-1, and then most coronavirus, and we have SARS-2. And then among the beta coronavirus, there's a subgroup, we call the SARS beta coronavirus or the SARS-Beta viruses. So these are especially kind of dangerous because we had the SARS-1 and SARS-2. And also because the nature they use the receptor ACE2 means that they can infect a lot of different animals. So we predict, currently we're worried about the next variant. So the SARS-CoV-2 variant through mutations. But there are viruses, in the natural reservoir and from intermediate hosts will jump out, and they will be the SARS-3 and SARS-4. So that comes to what's the natural reservoir and what's the intermediate reservoir host. We believe that bats are the major reservoir of the ancestral virus. So I will introduce the concept the ancestral virus versus the progenitor virus. The progenitor virus is the virus just before they jump to human and cause human disease. So bats are unique, flying mammals, and there are over 1,400 species. And we know for a fact now, at least 500 different coronaviruses have been detected that circulating in bats. The estimation is that at least, 5 times more than that. And so there are a lot of viruses in the bats, but bats only harbors what we call this ancestral virus. And for a virus to jump to human, almost invariably, they need to first jump into another animal we call the intermediate host. So the intermediate host serves for 2 purposes. One is to amplify the virus. The other is the evolution adaptation. So we believe that the viruses like SARS-1 and SARS-2 and the most coronaviruses clearly that have used the intermediate host to adapt and then eventually transmit to human. For SARS-1, we think is the civets, and for MERS, we think it is the camel. And for SARS-CoV-2, we're still searching for that one, So currently, in terms of surveillance sort of effort internationally, there's a lot of surveillance already ongoing, but most of them, I would say, by chance and by convenience. So what's lacking is a more systematic surveillance that to do larger scale and coordinated. So to do that, most people thinking funding is the most important. Funding is important, but I think technology is important. But to me, the most important is the political will and international collaboration. I always say fighting virus is like fighting a common enemy. So we have to cross borders and we have to have a very transparent international collaboration. So that's something, in order to develop a better vaccine, we have to know better about our enemies, and we have to have this transparent international collaboration. And then the gaps, and in terms of the future sort of surveillance efforts, first of all, we need to improve the international collaboration. And secondly, I think that I always say, I have been working with bats for a long, long time, but now I realize that surveying bats is not enough, because we're only going to know that population of the ancestral virus. That will help us, but that still is far away from the more accurate assessment of risk and spill over to human. So I think that the international collaboration and animal surveillance, genomic surveillance has to expand to the intermediate hosts. Of course, we don't know all the intermediate hosts yet, but we know certain intermediate hosts, for example, the civets, the raccoon dogs, the pangolins, mink, and a few others like the white-tailed deer in USA. So we already know quite a few wildlife animals are susceptible. And so my view is that in future, the surveillance should focus not only on bats, but also on this intermediate host. Last, I will finish up about these tools used for surveillance. Currently, the frontline tool is molecular. Either you do PCR or genomic sequencing. But I think, a serological surveillance, we need to develop smarter and more precision surveillance tools, because the antibody in the animals actually lasts longer. And also that will give us a much more sensitive approach and a broader picture about the prevalence and also the dynamics and the transmission patterns of these viruses. So thank you again. And I think that, it's clearly, very important that in the virology area and in the virus and the animal interaction area, we still have a long way to go to get the knowledge so that we can better advise the vaccine development. Thank you.
Thank you so much, Dr. Wang. I'll now turn it over to Dr. Barouch.
Well, thank you very much, Chris and Mike. It's my pleasure to be here today. It's also my pleasure to be one of many investigators who contributed to the coronavirus vaccine roadmap. Regarding immunology and immune correlates protection, we've learned a lot over the last several years, but there are important gaps in our knowledge for vaccine R&D that would be very beneficial both for SARS-CoV-2 vaccines as well as pan-coronavirus vaccines, and quite honestly, vaccinology in general. When the first mRNA COVID-19 vaccines emerged less than 1 year after the start of the pandemic, it was very clear that there was very unique immunology associated with these vaccines. They had very high early antibody responses against the ancestral virus and provided stunning degrees of initial efficacy. But problems with these vaccines emerged very quickly. One issue is that the durability of these vaccines was much shorter than initially anticipated or desired. The durability of these vaccines is measured in months, not years or decades. For many clinically licensed vaccines, a single shot is good for lifetime protection or only needing boosts after many years or decades. The rapidly waning immunity leads to the need for frequent boosting, which is logistically difficult and largely unpopular. Another problem that emerged is the rapid emergence of viral variants, which largely escape neutralizing antibody responses and decrease the efficacy of the vaccines. So that current vaccines do not provide substantial protection against acquisition of infection, but thankfully they still provide outstanding protection against severe disease. Thirdly, inflammatory properties of the vaccines lead to reactogenicity that dissuades some people from getting vaccines, and in some cases more severe complications such as myocarditis or thrombosis. And lastly, there's only limited cellular immunity induced by some of these vaccines, which is really necessary for long-term cross-reactive protection. Other vaccines that have been introduced, such as vector-based vaccines, have different immune profiles, including far lower levels of initial antibody responses, but higher levels of CD8 T cells and better durability. So it's clear that there is not yet the perfect vaccine. And immune correlates have shown that both antibodies and T cells contribute to protection. The field is focused like a laser beam on short-term neutralizing antibody responses, and while those are likely important, then the combination of humoral and cellular immunity working together, just like the immune system was intended, is likely critically important for coronavirus vaccines. In thinking for the future about next generation vaccines, these could be next generation COVID-19 vaccines that cover broad ranges of variants. These could be pan-coronavirus vaccines that provide protection against all sarbecoviruses or possibly even all beta coronaviruses moving forward. I would suggest that gaps in knowledge fall into multiple different areas. Of these, the 4 areas that deserve particular attention for vaccine R&D, including studying the durability of vaccines and how to improve durability. These studies could involve studies about antigen form and antigen expression. Secondly, we would need to have vaccines have better coverage of variants, both SARS-CoV-2 variants and Sarbeco variants, because it is very likely that variants will continue to emerge. So we need to have vaccines provide better cross-reactivity. Thirdly, we need to focus more on mucosal immunity. Mucosal immunity is a topic of high importance that has not received substantial attention during the pandemic. But having increased responses at mucosal sites is likely going to be very important for developing vaccines that block infection and block onward transmission. We know the current vaccines still provide good protection against severe disease, which is incredibly important, but the current vaccines really don't provide very good protection against acquisition of infection and mild disease. The hypothesis is vaccines that can induce better mucosal immunity would have a better shot at doing this. And finally, we need vaccines that induce better cellular immunity, because cellular immunity is the other side of the immune system, probably equally important to humoral immunity, and provides better cross-reactivity and durability of responses. To improve this, we need to improve existing vaccine platforms and develop new vaccine platforms, as well as focus on developing new antigens, both for SARS-CoV-2 as well as pan-coronavirus vaccines. To accomplish this, we really need a merging of basic research to understand these immunologic phenomena, coupled with applied research that includes both animal models as well as human clinical trials. And to accomplish projects of this magnitude, we really need to continue the collaboration between physicians and scientists, government and industry throughout the world. So I'll stop there. I'll emphasize that now is really the time to build the next generation of vaccines, and we need to do it now so that they're ready, hopefully before the next pandemic comes. Thank you very much, and I'll turn it back over to Chris.
Thank you so much, Dr. Barouch. Now I will turn it over to Dr. Weller.
Thank you, Chris, and thank you, Mike, Linfa, and Dan. You know, we've just heard about some of the gaps and needs highlighted in the R&D roadmap that will enable us to develop the next generation of vaccines. And I think what is clear is we have made a huge amount of progress, but there are still many unknowns and challenges and many challenges ahead of us. So really now is the time for us collectively to move the roadmap from a plan to implementing it. And some of that work has already begun. And I think there's 2 areas particularly that will enable this. Firstly, that continued investment, long-term sustainable investment in discovery R&D to advance the innovations and the basic knowledge, you know, structural biology, virology, immunology, and these are needed to develop the tools to be better prepared and to be able to act early. And the second piece is around the global coordination and global collaboration that we saw so keenly over the last few years. And Linfa really highlighted this in the surveillance. to have early warning systems in place, but importantly, not only to be able to see what's circulating, but also to link what viruses and viral variants are circulating with the R&D efforts, so we can see and then develop the right tools to control these variants and viruses before they escalate. However, I think one of our biggest challenges is one where we had the biggest failures in the pandemic, and that's around global access, which I'd just like to take a moment to speak on. You know, we all know that it took less than a year from the time the virus was first identified to authorization and distribution of vaccine. But there were huge global inequities in distribution and access of vaccines, particularly in low and middle income countries. And there were certain aspects that fueled this, including limited supply of vaccines. High income countries prioritize use of these vaccines for their own populations, understandably. Also, mRNA vaccines initially had a -80 cold chain requirement, which can make it particularly difficult to deliver these vaccines in certain parts of the world where there's limited electricity. And also manufacturing capacity was limited to high-income countries, or is limited to high-income countries, which means that these countries could control export and distribution, and therefore supply of vaccines around the world. And one of the themes that runs through the roadmap is the importance of integrating global access thinking and global access approaches, affordability, accessibility, and acceptability at the very beginning of the R&D efforts. So what is an example of what this looks like? Well, if you think about how a vaccine is delivered, and methods of delivery that may be less invasive and easier to use than injectable vaccines, such as microneedle patches. These can be placed on the skin, and these may not require the same health system support as injectable vaccines, and may not require the same cold chain requirements, so much more accessible around the world. But what remains critical to future vaccine R&D is to include the early and continuous engagement of countries and communities worldwide to enable uptake of future coronavirus vaccines once we have developed them. Now, in addition to the R&D efforts, I want to highlight just two areas where a concerted effort is needed to improve global equity in a more at a more systems level. The first is around regulatory capacity and capability, building and strengthening regulatory capabilities and systems in lower and middle income countries, which will be absolutely necessary for the review and approval of new broadly protective vaccines. And the second piece is around vaccine manufacturing, which is currently concentrated in high-income countries and a few middle-income countries. The pandemic highlighted the vulnerability of this position. For example, African countries waited many months longer than countries in the global north to receive vaccine doses. And this demonstrates the need to improve vaccine equity by increasing local supply of vaccines in Africa. And there has been much discussion about how to address this, with some very thoughtful suggestions centered around diversifying the geographical distribution of manufacturing capacity. But building manufacturing facilities alone is not enough. These facilities need to have business models that enable their commercial sustainability. And sustainability isn't just focused on the manufacturing facility. It includes the workforce, the expert workforce that is needed to run the facilities, but also for the supply chain, to provide the raw ingredients for the vaccines all the way through to the glass vials that they're produced in. And together, this collectively will enable robust vaccine manufacturing. So we need global coordination, global collaboration between industry, public health organizations, research organizations, and governments to move us closer to this goal. Now I think everyone here knows that we weren't prepared for COVID-19, and it's a fact that cost millions of lives and took a huge economic toll, and we can't afford to not be prepared again. Now is not the time to lose our focus. It's the time to really focus in and redouble our efforts and make investments in science to develop the solutions to protect our future. What we saw in the pandemic was that the research community was really galvanized. And I say research community in the broadest sense. It includes industry, public health organizations. The pandemic galvanized the research community, and through global collaborations, we were able to make multiple effective, safe vaccines. Time and again, we've shown that investment in science does bring new solutions. And now we must use this as the building blocks as the blueprint, build on this progress and get ahead of both new variants and future viruses before they begin to spread in humans. The R&D roadmap for broadly protective coronavirus vaccine provides us with a comprehensive plan and a structured path to accelerate R&D through improved coordination of concurrent and interdependent activities to deliver vaccines to everyone, everywhere. And with that, I'll hand back to Chris. Thank you very much.
Thank you so much, Dr. Weller. And now I'd like to turn it over to Dr. Nahid Bhadelia. Nahid, are you able to, we were having a little bit of technical difficulties earlier. Have we resolved that?
I hope you can hear me.
I can hear you.
It's been resolved.
It's been
That's great.
resolved. Thank you so much. I'll now turn it over to you. Thank you.
Thank you so much. And thank you for the invitation to talk a bit more in detail about Project NextGen and the important work that has been outlined by the roadmap and the points that this project would address. I don't need to make the case that has been perfectly made over the last few speakers about the necessity that we have to stay ahead of the rapidly evolving virus that is SARS-CoV-2 and the necessity of developing the next generation of tools because of that rapid evolution. While our vaccines are still very effective at preventing severe illness and death, they are less capable at reducing infections and transmissions over time. New variants and loss of immunity over time could continue to challenge our healthcare systems in the coming years. Similarly, the development of therapeutics that are effective against current and future variants remain a critical priority. An investment of at least $5 billion, collectively referred to as Project NextGen, will accelerate and streamline the rapid development of the next generation of vaccines and treatments through public-private collaborations. Based at the Department of Health and Human Services, and led by activities and efforts by ASPR's Biomedical Advanced Research and Development Authority, BARDA, and NIH's National Institute for Allergy and Infectious Diseases, NIAID, Project NextGen will coordinate across the federal government and the private sector to advance a new pipeline of innovative vaccines and therapeutics from labs to clinical trials to potential FDA licensure and commercial availability to the American people. This initial allocation of $5 billion for Project NextGen will be financed through ARP Testing and Provider Relief Fund resources, PRF resources. HHS has been able to reclaim those source resources through multiple contracts or executed at significantly less money than what the initial estimate was. Let me focus a bit more about the areas that NextGen will focus on. Mucosal vaccines that are delivered through the nose intranasally, or vaccines that induce mucosal immunity, which could have the potential to dramatically reduce infection and transmission, as Dan spoke about, in addition to preventing serious illness and death. Vaccines that provide broader protection against variants of concern and a longer duration of protection. Broader pan-coronavirus vaccines, which can protect against several different coronaviruses. More durable monoclonal antibodies that are resilient against new variants as they arise. And then advancing new technologies, to Charlie's point about how administration, routes of administration are an issue of global equity, advancing new technologies that improve access and enable faster, cheaper, rapid, and flexible production of vaccines and therapeutics. So the program has already started, and I'd refer you to the website, medicalcountermeasures.gov, to look at the BAA and the RFI that have been released by ASPR related to Project NextGen. The activities that are currently underway are including efforts that will bring the next generation of vaccines and treatments to American, but are not limited to supporting and advanced development efforts at BARDA to validate assays that can allow us to examine immune responses across new technologies, as well as further our understanding of biomarkers, in this case, correlates of immunity, that can predict the success of next-gen vaccines. It will also support existing NIAID clinical networks to create a platform for clinical studies capable of streamlining and standardizing the evaluation of multiple vaccine candidates and therapeutic candidates through consistency in data collection, clinical endpoints, and biomarkers. It will support public-private partnerships between BARDA and vaccine and therapeutic developers to make their products easier to bring through development pipelines. And these efforts will include advancing vaccine candidates with promising early-stage clinical trial data and bringing them to the next phase of clinical trials based on pre-established and standardized metrics. Evaluating next-generation monoclonal antibody products to meet the needs of our most vulnerable populations. GMP manufacturing of products for use in early-stage clinical trials, particularly vaccines utilizing new technologies and approaches. Lastly, it will support BARDA's development of innovative manufacturing approaches that will improve yields and will accelerate the availability of vaccines and therapeutics. Again, you can find the next generation related RFI and BAA information on medicalcountermeasures.gov. Building on experiences from HHS's COVID-19 response efforts over the last 3+ years, this administration will continue to pursue investments in a way that maximize the benefit for Americans and furthers overall scientific discovery. Project NextGen represents the continued commitment of this administration to address the ongoing threat of COVID-19 and to protect us against future epidemics from the same family of viruses that has been stressed by all the other speakers today that have caused 3 epidemics over the last 15 years, including MERS and SARS-1. And I will end by saying what's been referred to by Linfa and Charlie, the importance in this moment of global coordination to ensure that we can take a decisive step in reducing a risk from future coronavirus pandemics. Thank you so much, Chris.
Thank you so much, Dr. Bhadelia. At this point, I think we will go to some questions. If our speakers could go ahead and turn on their cameras. And the first thing I want to do is I want to thank each of you. The great remarks, really helpful, excellent high-level background material for people to think about. And as I mentioned earlier, the roadmap covers I think pretty much all of the issues that were discussed today. And please take a look at the roadmap if you want more detailed information on what our actual goals and milestones are that are outlined in the roadmap. So now we do have a few questions. And the first question, I think this is probably most appropriate for Linfa, but as we go through these questions, I think other people can feel free to jump in if you have any additional comments. So the first question is, what do we need to recognize pandemics early in their spread and to have the resources to contain them almost immediately? I mean, how do we do that? So Linfa, you talked a little bit about surveillance. So maybe we have you address this first, and then I'll see if anybody else has any additional comments on that.
Thanks, Chris, and thanks for the question. I mean, in terms of pandemic preparedness, I always say the highest hanging fruit is to do the surveillance. And as I said, in the reservoir host, the intermediate host, and hopefully we have enough knowledge to risk assessment and prevent the spillover to human. So that still is a dream. I think, we're trying, but failing that, the next step is early warning. So that's where I think the surveillance in human population, especially the ICU unit. So I think usually, I have been doing this for the last 30 years. There's 2 things can trigger early warning. One is a very severe case of unknown etiology, and then the patient had some kind of animal contact. So then it can really indicate there's a new zoonotic disease. The second is a cluster of diseases, may or may not have a severe, but uncommon phenomenon, symptoms, and you don't have an etiology, and then you have some epilink to animals. So these are the 2 areas that I think is that human sort of hospital-based surveillance or community-based surveillance, and we can steer if we detect that, and we can steer respond early. Of course, to do with that, then we have to have the countermeasures. So this is the vaccine, the NextGen vaccine. If we do this early warning, plus in our countermeasures, we have the vaccine, we can do ring vaccination, or we can have therapeutics to dampen the transmission, then that's the third sort of a layer of defense we have. So it's really the prevention of the spillover. If that doesn't have not be done, early warning in the severe case in the early cluster. But to do that, then we need to do containment. So one of the containment methodology is ring vaccination or using broad antiviral therapeutics to dampen the transmission. Thank you.
Thank you.
Chris, can I just add to that, because this has been a topic near and dear to my heart for a better part of 30 years. And I agree with everything Linfa just said, that in the ideal world, we could avoid the spillover or detect the spillover quickly and contain it. My sense from a reality standpoint is that once you have a virus with wings, a respiratory viral pathogen with relatively moderate to high infectivity, it's gone. In 2009, in the first month, from the time period that we first recognized H1N1 in Mexico in humans, it was in 128 countries in 1 month. If you look at the retrospective on SARS-CoV-2 and whatever the debate might be about when it emerged, where it emerged, if you saw how quickly it moved around the world. And so ideally, the countermeasures would stop it cold in its tracks. But more importantly, I think if that doesn't happen, which I think is more likely to the case, this is why vaccines become even more important. We can't wait to figure out what vaccine we're going to develop when the pandemic begins if we're really going to have an impact on it. That's why I think the research and development that Dan laid out that we've heard from is really, really important to have those vaccines in pre-deployment potentially. Wouldn't it be ideal if we could take a pandemic off the table by having stopped it from ever spilling over because of a vaccine program was in place? But if that's not the case, at least how quickly can those vaccines be moved? And I think what we've learned from these viruses, even 100 days, which is clearly what we're talking about today with the CIPI's efforts and so forth, to have vaccines out. And as Charlie laid out, to deliver them is a whole nother time period beyond having the vaccine to begin with. That virus will fly around the world. And so I think this is why, for me, vaccines take on even more importance now than ever in terms of either early recognition or at least once it happened, trying to protect the world as best we can from its ongoing transmission.
Thank you, Mike. Anybody else want to add anything to what Mike and Linfa have said? Charlie?
Yeah, I just want to actually, Mike said what I put my hand up to say exactly the same, but I think it's a reiteration of what Mike was saying about that link between the surveillance networks and early warning systems and then a responsive medical countermeasure environment. And I think it's a little bit of a shift in our thinking about how we do vaccine development and shifting more of the work into the preparedness space and how much can we do before, and that preventative and preparedness frame of mind, so that when something does pop up and we have the early warning systems in place and the surveillance working beautifully, we have somewhere to go. We're not starting from scratch. And that might also mean that the clinical trials that we know today, we might have to rethink how we do those and maybe what we can do later as well. I'm not saying that we cut corners or anything. It's more how to think differently about how we manage clinical studies and in a pandemic or in response. Over.
Great. Any other comments on this? A related question also maybe for Linfa is you mentioned the importance of trying to detect these spillover events early or before they spill over. Are you aware of surveillance in animal populations for coronaviruses, and specifically surveillance towards these intermediary hosts? And what do you think, first part, and then secondly, what do you think would be what would you recommend in terms of conducting this kind of surveillance? What is needed to try to detect these viruses before they spill over? Could you comment on that briefly?
So as I sort of mentioned in my introduction, clearly that the technology and the funding need to be really well advanced and coordinated. But personally, I have been doing this for 2 decades now. I found that ironically, COVID-19 made it even more difficult. So the country's willingness to let you to survey and to share the information, that to me right now is the biggest stumbling block. So that, I think that's what I said, need the political will of the national leaders, the international leaders, and also some legal framework, right? So that, virus recognize no borders. And as people nickname me as a Batman, I always say, Bat recognize no borders. So that if we don't have a legal framework to say we don't care where the virus is from, if we detect that, the international community will all benefit. So that legal framework and the political will has to be there. Then the funding and the technology really, to me, is secondary right now. I used to think, as a scientist, create some surveillance tool, we can solve all the issues. But I now change my mind to say, the political will and the transparency and the legal framework is even more important than the funding and the technology. Thank you.
Okay, and this is kind of a related question. I think this is actually a little bit organized towards human surveillance, but the question is, could the speakers address the challenges of building a global viral surveillance system? What do you see are the barriers we would need to overcome? And maybe Linfa, why don't you respond? And then Charlie, if you have any thoughts on this, please go ahead.
I thought that the issues would be similar, right? So Charlie might be in a better position in terms of the human system that it's the reporting. To me, it's the reporting and also what triggers alarm bell to go to WHO and then take necessary containment or investigation. Send missions to the country and things like that. So Charlie, I'll pass to you. Yeah.
I think you've already mentioned, Linfa, about the political will, and sharing of information is one of the major barriers. I think if we, from what we've learned in COVID, what went well, what didn't, and how can we build the right frameworks for inevitably, which is an on-ramp or off-ramp or switch, whatever the trigger is to start the process. At the moment, we have FEIX, public health emergency of international concern, as one trigger. Whether that is too late, whether there should be something beforehand, a rethinking of what steps beforehand can give us an early indication. And I think along those lines is maybe also a rethinking about risk and being more comfortable with starting earlier and taking early risk, and hopefully nothing will come from it. But that is a risk in starting earlier before we know what the situation is. Over.
Nahi, do you have your hand up? If you want to add a comment, please do.
Yeah, I do. I just maybe because first and foremost, I am a frontline healthcare worker before anything else. I wanted to point out the importance of our investment in healthcare worker and healthcare workforce, which is why one of the important facts and points of the U.S. support of the pandemic fund has been holistic disease surveillance and workforce development. This is such an important part that gets missed. You need somebody to be able to diagnose those patients, and then you need somebody to be able to upload those sequences. So just wanted to put that point.
Great. Any other comments on this issue? Okay, the next question is, how will obligations to make the resulting products equitably available be embedded in the development agreements and processes? Nahi, this may be something that you can speak to, and then I don't know, Charlie or Mike, if you want to speak to this as well. Any comments on that?
Yeah, I will just say that one of the ways that the approach to Project NextGen is going to go is to take the lessons that were learned from the last 3.5 years, right, from the development of these the initial vaccines, to see how we can not just about ensuring that we have the maximum yield for American public, but also that we are doing that work in a way that supports further scientific discovery and collaboration. So I will stop there and actually probably refer you more to BARDA and ASPR for details about how they're going to approach that.
And Charlie, do you have additional comments on that?
Just a very short one, that this is tough, and CEPI has done some work towards including some access agreements as part of their work over the last few years, and so learning from they've evolved and they've been learning from their experiences, but I think this is a work in progress that needs more attention. Over.
A kind of a related question, and I think maybe Charlie for you as well, is can you speak, you mentioned the need for manufacturing capacity and expanding manufacturing capacity globally. Can you speak to any ongoing efforts that are kind of trying to make that happen? I mean, what is happening in that space to try to improve manufacturing capacity on a global level?
Yeah, no, it's an important, it's all very well stating what the problem is, but without a solution. And so it's a long-term process building capability, because it's about people as well, and the workforce that was mentioned just earlier. And there's the WHO mRNA tech transfer hubs that are happening in tech transfer, connecting organizations that have a huge amount of expertise with other organizations, manufacturing organizations that may have less. And some of the work that we've been doing is building that expertise by doing tech transfers, not necessarily on coronavirus, but it's in the area of different platform technologies for vaccines. And so this is, it's a long-term process, and also the coordination between different manufacturing organizations to understand where each other's strengths are. And that's tricky because, these are commercial organizations. Over.
And Mike, did you want to add anything there? Well, Mike, you're on mute.
Yeah, I think that the line of questioning here is really very critical. But to me, it's the whole idea, you creep, you walk, you run. And what we're talking about is how to actualize our running. And I'd like to maybe back this up a little bit to talk about our creeping and then walking. And maybe this is really aimed to Dan and Nahid, is the fact that in an ideal world, what would be the timeline? And Dan, your experience, obviously, across vaccine development and immunology, et cetera, how far are we away in your best professional judgment, without anybody holding you to it, from one of those vaccines? And that we could surely see as game changing. And then, Nahid, I think it really spills over to your area, of which, by the way, thank you for your government service, having been plucked out of academia to come and do this. That takes a lot of effort and commitment, and I appreciate that.
Yeah.
So you're not answering this as a career politician, but as a scientist who's been exposed to this. How do we maintain the time period of funding to get to that endpoint? Because I'll be surprised if Dan tells me that at the end of 18 months, we're going to have the game-changing vaccine. And what I worry about is that we're not setting our sights on them. One of the challenges I think we had with the early days of the current vaccines is after the initial antibody responses and those first 2 months of protection, we kind of more or less gave the public the sense this was a 2-shot and out deal. And then when that didn't happen, we began to lose a lot of credibility. And so I'm trying to help us lay out a realistic credibility timeline that says how long to get to one of these vaccines if we can, and how will we sustain government funding? So Dan, maybe if I could ask you that, you think about a lot about the ideal vaccine and what might be achievable.
Sure, thanks, Mike. So, I don't think that it's not just a black or white game-changing vaccine. I think what we will see is essentially reflective of the natural progression of science, which will be iterative improvements. Occasionally, there will be game-changing technologies that come to bear, but then there will be limitations of those technologies as well, regardless of what those technologies are. So I do think that we will see gradual improvements of different technologies in terms of vaccines that don't require cold chain, vaccines that have increasing durability, increasing mucosal immunity. And I think that what we'll see some advances if adequately resourced, certainly within 18 months, but I don't think that will stop there. I think it'll be a scientific process over a number of years to really improve vaccine platforms to the level that we're talking about today. Also, I fully agree with you that the communication about vaccines needs to be done very carefully to ensure that outstanding performance, say 2 months after initial vaccine, we know all too well that may not actually pan out to be that level of efficacy in the long term.
Nahid, do you want to add
Mike,
anything? Yeah, go ahead.
Sure. I will follow Mike's point by saying the number one lesson I've learned when you switch over from academic to government is that you never take the government hat off. So I can't quite take the government hat off. But here's what I have learned. Investments, like the government investment in Project NextGen, have the power to do a couple of powerful things. They have the power to move potential ideas and projects that there may not be market forces to move more quickly. They can reduce the time to potentially having new technology come to market in a way that they may not have. And third, I think an important point that was made to me recently as we were thinking about this is there's so much intellectual capital and brain trust right now on folks that are working on SARS-CoV-2. And this investment, such as Project NextGen, can retain that brain trust in those companies, in those academic institutions to continue to work on that. And that's the third part in which that project investment like the NextGen could help. The last thing that I'll mention is that you heard the kind of activities that may occur, that are occurring rather, I should say, under Project NextGen. The ability to create standardized assays or effective assays to measure how we will measure the efficacy of future technologies, the ability to create a standardized clinical trials platform, all of those things are also ways in which you might be able to reduce the timing and potentially test multiple products, which I think can actually help the time.
Very helpful. So the next question, and this is kind of related, maybe we've sort of answered this, but it says, much of this represents a paradigm shift in how we traditionally have approached vaccines, and rightfully so. How do we educate the regulators and the general public for our best chances of success? Anybody want to, we've kind of talked a little bit about this, but any other additional comments, maybe particularly towards regulators and what is needed from a regulatory perspective?
I can comment very quickly. I think, a theme of what we've been talking about within the roadmap is that coordination and bringing different sectors together. So including regulators early on in any new technologies, any new developments, so they have time and chance to get their heads around kind of these new technologies is always going to be helpful. And I know that I've heard regulators say, please include us early. And I think the other aspect is strengthening regulatory expertise and systems across the world so that there is equivalent regulatory expertise everywhere. Over.
And I think, can I just add to that?
Please.
I think that the roadmap process really was all about that. I mean, I sat across the screen from regulators, scientists, public policy experts, funders, who brought together the priorities for this roadmap. And once you understand common priorities, then you can begin to understand, now, how do I need to address these from my discipline, from where I'm at? And I'm more optimistic that this will happen with the roadmap type process, where it did have such stakeholder involvement. And this wasn't an immunologist report. This wasn't a regulator's report. This wasn't a funder's report. This was a collaborative effort report. And hopefully that will help spur on this kind of coordination and very much support the point that Charlie just made. I agree with her 100%.
Yeah, and I think, so we've got a couple minutes left. And I think the last question that we may want to deal with is, so even though COVID-19 continues to cause severe disease and death worldwide, it seems like the world has kind of moved on. And we very much appreciate this effort by the US government to do the NextGen project, and that is really fabulous. But how do we, other than that effort, how do we continue to build advocacy and the political will for continued investment? But it's not just investment. It also gets back to what Linfa was saying about, the need for this kind of collaboration, the need for transparency. Yeah, how do we take what we've learned and build that for the future to ensure that we can continue to do this kind of effort in these researchers? Any research, any comments on how do we get that advocacy piece better known out there? Mike, I'll turn to you. You're always good at this kind of stuff. And then
I'll go as well. Yeah.
No, I'll let you go first if you'd like, Linfa. Go ahead.
Well, I think in the context in Singapore, in the region, I say governments are very used to military preparedness, economic preparedness. I think that we need now pandemic preparedness at that level, basically. Thank you.
Yeah, and I was just going to actually say very similar words. When we think about a pandemic, so often our response is time to when the pandemic emerges. And remember, we were very fortunate with the mRNA technologies, for example, that they were never meant for responding to a pandemic, but they very fortunately provided a very important platform. You don't build an aircraft carrier the day you go to war. You build an aircraft carrier 15 years before, and you start that process. And I think, as Linfa just very thoughtfully said, we need to adopt that kind of concept right now. That we're not just talking about endemic disease, which is important, You know, if you look at between pandemics, a lot of people still die from these diseases that are not part of the pandemic. But when the pandemic happens, that's not the time to build your vaccine aircraft carrier. And I think that's what we're really needing to get the point across, is how that investment is so important. So when the pandemic happens, we are in that much better prepared mode.
Well, I think that's a great last word and a great place for us to stop. We're just about at the top of the hour. And again, I want to really thank our
Thank you.
speakers today, and particularly those that were involved in our task force and their really robust input and support of this project. And Mike, thank you for your leadership on this. And I
Thank you.
wish you all a good rest of your day, and thank you, everyone.
Thank you.
Thank you.
Thanks,
Chris.
everybody.
Thanks,
Thank
everyone.
you, everybody.
Bye.